nervous system. We have axons that travel together in the central nervous system, but they're not called a nerve, it's called a tract. Okay, so what is a tract? It's exactly the same as a nerve, except it doesn't have those connective tissue coverings that we talked about. There's no epineurium, perineurium, endoneurium. It's just a group of axons traveling together in the brain or spinal cord. So notice that we have different types of tracts depending on the direction that the axons travel. Okay, so if the axons cross over the midline, connecting the right and left sides of the brain, this is called a commissural tract. Okay, commissural tract. The largest commissural tract in the nervous system is something you guys are going to see a lot because it's very large. It's called the corpus callosum. Okay, corpus callosum. You'll see this in just about any cut in the human brain. Okay, you'll see it in the sheep brain. So if you're looking at a mid-sagittal cut, it's actually this big kind of C-shaped structure here. So these are actually axons that are crossing over the midline, connecting different parts of the right and left side of the brain, allowing them to communicate with each other. Okay? So commissural tracts cross over the midline. Okay, if the axons are not crossing over the midline, but they're connecting parts of the same side, this could be the same side of, like connecting, for instance, the frontal lobe to the occipital lobe, or just going from one, little gyrus to the next. Those little hills are called gyri. So these could be very short, or they could be longer. If they don't cross over the midline, they are called association tracts. Okay, so these would connect parts of the same side of the brain. Okay, and then finally, if the axons are traveling vertically, they could be connecting like a higher center to a lower center in the brain, or maybe connecting the brain to the spinal cord. These are called projection tracts. Okay, and these can be either carrying ascending information, meaning it's traveling up. What type of information would it be if it's ascending? Sensory or motor?
Sensory.
Sensory, right. Or they could be descending, where the information is traveling down, and that would be motor. Right. So these are just general terms based on the direction that the axons are traveling, right? Okay. You guys do have a diagram that illustrates this, so I will show you that in just a minute. So this is the terminology in the central nervous system. Okay, a group of neuron cell bodies is a nucleus. A bunch of axons traveling together is called a tract. Okay, so they like to make everything different. And so, of course, in the peripheral nervous system, we have different terminology. And actually, I think we saw this term last time also. A ganglion is a group of neuron cell bodies located together, but out in the PNS. Okay? And then a nerve, again, it's a group of axons traveling together. That's all there is to it. Okay? Okay, so I think we're going to see some diagrams showing this stuff. A couple more terms. We talked a little bit last time about how myelin is what forms our white matter. So what is gray matter? Well, gray matter is everything that's unmyelinated. So this would be our neuron cell bodies. It would be dendrites, because they are not myelinated, and any unmyelinated axons. So that is what forms gray matter. Okay, white matter is all of the myelinated axons. Okay. Okay, so this is a table that gives you some of these definitions and taking a look at some cuts through the brain. So these are different parts of the brain as we go from the cerebral hemispheres up here. This is actually a frontal section through the cerebrum. This is farther down in the brain stem. Again, down in the brain stem. And we'll talk about these areas in just a minute. And then this is a cut through the spinal cord. So notice that really no matter where we make these cuts, you can see how the gray matter, which looks kind of a darker tan color, and the white matter is kind of separated. Okay, so that means all of this stuff here would be like neuron cell bodies, dendrites, maybe we'd have some unmyelinated axons, okay? And then all this stuff, all the white matter, would be myelinated axons that are traveling, through and connecting different parts of the brain or spinal cord, okay? So let's take a look here, and just because we've talked about some of these terms now, actually the outer few millimeters of the brain's surface is where we have most of our neuron cell bodies. This is where anything that's on a conscious level is happening, meaning like what we're able to consciously think about. Like right now you're hearing the sound of my voice, and hopefully you're thinking about what I'm saying, or whatever you happen to be thinking about is happening somewhere in this area. We have billions and billions of neuron cell bodies here. This is called the cerebral cortex, okay? It's the outer just few millimeters of tissue, but contains billions of neuron cell bodies. So it is gray matter. These areas here also gray matter because it's a bunch of neuron cell bodies. These are actually nuclei, okay? So we just talked about the term nucleus, right? Nuclei is plural, as you guys know. So these would be just clusters of neuron cell bodies, okay? Now, of course, they all have their own specific names, but they are nuclei. They're groups of neuron cell bodies. Okay. This actually happens to be the corpus callosum, the tract that we just talked about. So why does it look different here? Because we were looking at a mid-sagittal cut before, and this is a frontal section. But notice in this frontal section, you can see how the axons would connect the right and left sides of the brain because they go across this way. Okay, that's what they would look like. So allowing the two sides to communicate with each other. Okay, so this is what we're going to be talking about in just a few minutes. We're going to start with these spaces that are inside the brain. These are what are called the ventricles. And notice that when you make these different cuts, you can see the ventricles. Okay, so that is what we're going to talk about. What would be inside here? Again, cerebrospinal fluid, right. Okay. So before we start talking about the brain, let's just kind of talk about the major regions of the brain so that we are kind of, familiar with the parts, the main parts of the brain. Do you guys have any questions before we go on? Okay. Oh, and just coming back one more time to those different tracks. Remember we said that tracks that cross over the midline, shown here in blue, this actually happens to be the corpus callosum, but tracks that cross over the midline are called commissural tracks. Okay, tracks that travel vertically, like these green ones, are called projection tracks, and yeah, they are labeled here. And then tracks that do not cross over the midline but connect different parts of the same side are called association tracks. All of these shown here are association tracks. Okay, so this is just illustrating those different types of tracks. And again, these two are both association. Just kind of breaking them down into the longer and the shorter, you don't have to know those terms. Okay, questions? Yeah, in this diagram here, this is showing one half of the brain, and it's showing short association tracts, which are called arcuate because they kind of have that arc shape, and then the longer association tracts, which are called longitudinal, and you don't have to know those terms. They're all association tracts. That's all you need to know. If they don't cross over the midline, they're association tracts. Okay? And the green, because they're traveling vertically, are projection tracts. The blue would be, again, anything that crosses over the midline is going to be a commissural tract. But this one here actually happens to be the corpus callosum. So that is a specific name for this commissural tract. Okay. And you can actually see another ventricle here in this picture. Do you guys have any other questions on this before we go on? What would be vertical?
The midline would be a mid-sagittal cut?
Yeah.
Across the midline, so the midline is mid-sagittal.
Yeah, so this is a mid-sagittal cut. This isn't showing the cerebellum or the brainstem, so it looks a little different than you see here, just because it's really just showing the cerebrum. It's just showing that part of the brain. But this is a mid-sagittal cut, and this is a frontal or coronal. Okay. You guys have any other questions on any of that? Okay, so let's just name the parts of the brain, and so we can get started going through them and talk about them in a little bit more detail. In humans, this area is what has become really huge compared to other species. So you'll see in the sheep brain, although the sheep brain has the same parts that we have, their cerebrum is much smaller. And so these are called the cerebral hemispheres because we have two, a right and a left cerebral hemisphere, or you can just say cerebrum. There is a fissure that travels down the middle, which you guys need to know, so I'll try to show you that real quick here. These smaller kind of crevices on the surface are called sulci. You can say sulci, some people say sulci, technically it's supposed to be sulci. So singular is sulcus. So each of these little crevices is a sulcus. Okay, so again, sulci would be plural. A fissure is a deeper sulcus. Okay, and so why would we have these little hills and crevices on the surface of the brain? What would that do for it? Increase surface area. So remember we said in the outer few millimeters, that is where most of our neuron cell bodies are, right? So this increases the surface area for the cortex. So we have more neuron cell bodies there. Okay, so notice here, we have a really deep fissure. And I think they actually forgot, unless they've added a label in the new edition, they forgot to label this. They mention it in the text, but they forgot to label it. So you can add this label somewhere. This is right here. This is called the transverse fissure. Okay, because it's in the transverse plane. Okay, so notice it is between the cerebrum and the cerebellum, which is this part of the brain right here. Okay, so it would be right there, the transverse fissure. Right there. Between the two cerebral hemispheres, and I'll try to hold these together without dropping everything. This deep fissure that travels down the center, okay, right here, is called the longitudinal fissure. I don't know why it's not called the sagittal, but it's not. It's called the longitudinal, okay? So that is this one here. And you need to know those fissures. We will talk later about a couple of these sulci that I would like you to know the names of, but we'll come back to that. Okay, and the little hill-like structures, the little elevations here are called gyri. So one is a gyrus, plural is gyri. And again, those would just increase surface area. Mm-hmm. We'll actually, we'll be talking about it later, but you guys see, let me use this picture here. You can see there's a sulcus here coming around the temporal lobe. This is the temporal lobe. So there's a sulcus here. If you pull the temporal lobe back and look inside that sulcus, that is where the insula is located. So we'll talk about all of the lobes. It's actually considered to be a fifth lobe of the brain. We'll talk about all the lobes in just a little while. Okay, so I got kind of sidetracked there with those fissures. Let's come back to these parts of the brain. So again, this is the cerebrum, and the cerebrum sits on top of an area called the diencephalon. You actually can't see it here because it's kind of up inside. It's a little farther up, and the cerebrum kind of covers it. So let's take a look at a mid-sagittal cut. This is showing the inferior surface of the brain, and notice this is actually where all the cranial nerves are connected to the brain. So all of these nerves that are exiting from the brain, you guys are going to be learning. These are called cranial nerves. We'll come back to them later. Okay, so this is a mid-sagittal cut. So again, here we have the cerebrum. This is one cerebral hemisphere. This is the corpus callosum, that commissural tract. So axons would be coming out toward you because they're crossing over the midline. Okay, here we have again the cerebellum. The cerebellum is an important part of the brain for motor coordination and balance. It actually, the name means little brain, and it kind of looks like a little miniature cerebrum. And this again right here is the transverse fissure. Okay, so right below the cerebrum, this area here is called the diencephalon, and I will outline it for you. Okay, so that is the diencephalon. And then everything below that is what makes up the brain stem. So the brain stem is this area here. So I'm going to put these in a different color so you can kind of see them. This is the midbrain. Okay, it's only about an inch or so long. This kind of big anterior bulge here is the pons, and then below that, this area, so the pons would be right there. This area below that, again, it's only about an inch or so long, is called the medulla oblongata, usually just called the medulla. And you can see the medulla is continuous with the spinal cord. The boundary between them is actually the foramen magnum. So you guys remember the foramen magnum. When this travels through the foramen magnum, then it becomes the spinal cord. So these three areas here, the midbrain, which is also called the mesencephalon, the pons, and the medulla, together make up the brain stem. Okay? So we have cerebrum, diencephalon, midbrain, pons, medulla, and then the cerebellum. Notice that the cerebellum is not part of the brainstem, it's posterior to the brainstem. Okay? Okay. So now we're going to talk about those spaces inside the brain which are called ventricles. Notice that in certain cuts, you don't really see the ventricles. This is one of those cuts, this mid-sagittal cut. You can't really see them when you're looking at the diagram. And so the best place to see the ventricles would be either on the model that shows them, okay, which is this. I know this looks weird, but this is exactly what the ventricles look like. This material, this plastic stuff, is actually poured into a human brain, not while the person's alive, but a real human brain, and then they remove the mold once it hardens. So these are actual, like this is exactly what the ventricles look like, made from a real human brain. And you can also see them on one of the diagrams, which we will take a look at now. Do you guys have any questions on any of this before we go on? Okay. Moving on. Okay, so this is a description of the ventricles, but I would like to show you these looking at the actual diagram. Okay, so notice that this looks exactly like this model, okay? You can see the shape and position of them here. So this is what is called a phantom view, like we're looking through the tissue of the brain, and you can see the shape and location of the ventricles. Okay, so first of all, in each ventricle, there is an area called the choroid plexus. And that is what actually makes cerebrospinal fluid. Okay, choroid plexus. it makes CSF. Okay, so I'm just gonna abbreviate cerebrospinal fluid. We talked about this a little bit last time. We said that the ependymal cells, you guys remember ependymal cells, right? Just mentioned those a little while ago. Those are the neuroglial cells that line the ventricles. It's simple cuboidal epithelium with cilia. So the choroid plexus is made up of the ependymal cells that line the ventricle plus the underlying capillaries, the underlying blood capillaries. Okay, so we have little capillaries, which are the tiniest blood vessels in the body. And normally what happens in our blood capillaries, and this is true for the entire body, is a little blood plasma will leak across their wall normally. This happens everywhere in the body, and that's what carries nutrients to all of our tissue cells. So as the blood plasma leaks across the capillary walls, it will go through the ependymal cells into the cavity of the ventricle. And once that blood plasma is in the cavity of the ventricle, it becomes cerebrospinal fluid. So cerebrospinal fluid is actually very similar to blood plasma. It just comes from the blood. It will circulate through the ventricles very slowly, and it will eventually get reabsorbed back into the blood. So it makes like a complete circuit, okay? So we're gonna talk about how cerebrospinal fluid does this. And we'll trace it through the ventricles. So each ventricle has a choroid plexus somewhere in its wall. Okay, so first of all, the lateral ventricles, these two here, are just called the lateral ventricles. They are located in the cerebrum. Okay, I would like you to know the locations of these. So these are located in these cerebral hemispheres. And you can see these are really large. Because they are big, they make about 80% of the total volume of CSF. Okay, so notice the lateral ventricles have these weird projections that stick out. These are called horns. The anterior horns are in the frontal lobes. And the lobes, I know we have not talked about those, but you guys probably can figure them out because these are named like the bones, right? So these would be frontal lobes. What would these be up here? Parietal. Back here they would be occipital, and here they are temporal. Right. Okay, so these anterior horns are in the frontal lobes of the cerebrum. The posterior horns are in the occipital lobes, and then these are called inferior horns. They're down in the temporal lobes. Okay, and I do want you to know what these horns are called. Okay. So cerebrospinal fluid is going to circulate through all of the ventricles. They're all connected. So CSF will go through these two little connections here. Can you see those little connectors? Okay, so these will go from each of the lateral ventricles down into this ventricle here. Okay, so CSF goes through those little connections. These little connections are called the interventricular foramina. Interventricular foramina, okay? Interventricular meaning between the ventricles. Okay, so when you guys are looking at this model, these are obviously lateral ventricles, right? This whole thing, anterior horn, posterior horn, inferior horn. These little connectors right here, you can see, I know it's hard for you all to see what I'm pointing to. You guys can move up if you want. These are the interventricular foramina, okay? It's a little connector that goes between the lateral and the third ventricle. So these little guys here. Okay, so we think of a foramen as being a hole, right? So when you look at this, you want to try to remember this whole thing. These are a bunch of holes, really. These are spaces inside the brain. So when you look at this, you don't want to think you're going to find a hole on the model that's going to be the interventricular foramen. It doesn't look like that. It's this little solid structure that connects the lateral to the third ventricle, okay? These little connectors. Okay, so the next ventricle is called the third ventricle. Don't ask me why. These are not numbered, they're called lateral, but this one is called the third. This is in the diencephalon, that area below the cerebrum. Okay, the third ventricle is in the diencephalon. Now notice the third ventricle does have what looks like a hole in it. Why is that there? This is because it is right in the midline, and actually there's a piece of tissue that travels through there connecting the two sides of the brain in that area. Okay, so we'll talk more about that later. Okay, the next structure here is actually called the cerebral aqueduct. This connects the third and the fourth ventricles. So this skinny little channel here is the cerebral aqueduct. This travels through the midbrain. Okay, cerebral aqueduct travels through the midbrain. You can call this the midbrain, or you can say mesencephalon. I do not care, it's the same thing. Okay, so it is the cerebral aqueduct. That will carry the CSF down into the fourth ventricle. Now remember that each ventricle does make more cerebrospinal fluid, okay? So each ventricle has its own choroid plexus, and they're all connected. So the cerebrospinal fluid just kind of circulating through all these areas. It gets down into the fourth ventricle. This is the smallest ventricle. This is where it's going to leave the ventricles now, because the fourth ventricle has 3 openings that are called apertures, okay? So it has 2 lateral apertures, okay? You can see that name here. 2 lateral apertures and 1 median aperture. Okay. So the CSF leaves the fourth ventricle through the 2 lateral apertures and 1 median aperture. I think that the reason they don't call these foramina is so that you don't get them confused with the interventricular foramina, okay? So you can kind of see where those apertures would be on the model, but I will not ask you to identify them on the model. But you need to know this. This is important because this is what allows cerebrospinal fluid to now leave the ventricles, and it's going to go into a space where it will now circulate around the brain and the cord. And that space is called the subarachnoid space. Okay. Okay, so to understand where the subarachnoid space is, we need to talk about the meninges. Do you guys feel okay with the ventricles?
I need to go over the process from the beginning, like
Kind of the
travel.
flow?
Yeah.
Okay,
But
so
I could do that later on. You better go over it.
we'll put it all together, actually. Okay, after we talk about the subarachnoid space, we'll put all of this together. And we'll follow their cerebrospinal fluid from the ventricles out into the subarachnoid space, and then look at where it's reabsorbed into the blood. Okay, other questions? Anyone else have questions? Okay, so this is actually a drawing of what the choroid plexus might look like. I don't know if this is the most helpful drawing in the world, but it shows a blood capillary, and it shows again how the blood plasma leaks out across the wall of the capillary. It goes through the ependymal cells, and now it's out in the cavity of the ventricle. Once that blood plasma is in the ventricle, we call it cerebrospinal fluid, okay? Oops. Cerebrospinal fluid. So here it's blood plasma, but here, because it's in a different space, we call it cerebrospinal fluid. It's very similar to blood plasma in composition. And this is an actual brain. It's a cut through a brain, and you can see the choroid plexus here. It looks like just kind of some I don't know, doesn't look like much, but it's blood vessels. Okay. Alrighty, so the meninges. These are membranes that cover the brain and the spinal cord. They are basically the same around the brain and the cord with one difference. This outer layer is actually a double layer around the brain. Around the spinal cord, it's just a single layer. Okay, so we'll go through these. You guys have heard of these, right? What is inflammation of the meninges called? Meningitis, right, you've all heard of meningitis. Okay, so these are important to know about. The outermost layer of the meninges is called the dura mater. This is a very tough, fibrous, connective tissue. It's dense, irregular. You will see, if you get a sheep brain with this attached, you'll see it's very tough. It's actually hard to cut. This literally means tough mother. It really does. I did not make that up. Okay, so around the brain, it has two layers. Around the spinal cord, it's just one. Okay. So because it has just one layer around the spinal cord, we just call it dura mater, around the cord. But around the brain, the two layers, of course, have to have a name. Okay, so the outer layer is going to be right up against the bone. You guys know the periosteum of the bone. Remember that? It's like that connective tissue membrane that covers bone. Okay, so the outer layer is called the periosteal layer. This is up against the bone of the skull. The deeper layer is called the meningeal layer. Now this is going to do some funky things because it's going to come down into those fissures, okay, that we just talked about. And this just kind of helps to wrap around the brain, giving it a little bit more protection and cushion, okay? So where that meningeal layer comes into the longitudinal fissure here, it is called the falx cerebri. Okay, cerebri because it's the cerebrum. Falx means it's like a C shape. I know this word doesn't really look like that's what it means, but it means it's a C shape. Okay, this means like sickle shaped. You know what a sickle looks like? Okay, so this is between the cerebral hemispheres. I just happen to have a model that shows it. Okay, so this is exactly what this model looks like. There is no brain for this model. This is the way it's supposed to be because this is supposed to show you the parts of the dura mater, and that's what this is. So you can kind of see how this would form a little shelf of tissue between the two cerebral hemispheres, right? Kind of helping to hold on to them and protect them. Okay, so this is the falx cerebri, this guy right here. Obviously, it's not really plastic in our bodies. It's dura mater, but it is very tough. The periosteal layer would be this, right up against the bone. This is bone. It's supposed to be bone. Okay, so where it comes into the transverse fissure, remember the transverse fissure between the cerebrum and the cerebellum? It forms this structure. This is called the tentorium cerebelli. The cerebellum would be right down here. The cerebrum is up here. Okay, cerebrum, cerebellum. So this separates them. Tentorium, like a tent. Okay, tentorium cerebelli. Sorry, I know that's way down there. Tentorium cerebelli. So that is in the transverse fissure. The falx cerebri is in the longitudinal fissure between the cerebral hemispheres. Now, the longitudinal fissure, it extends down here between the cerebellar hemispheres also. So we have a little piece of dura mater that comes up right here. It's very, very short. You have to look at the base of the model here to see this. You'll see it's very short. There's a little piece of dura mater here that is called the falx cerebelli. Okay, so it's basically the same as the falx cerebri, but it's between the cerebellar hemispheres. Okay, so falx cerebelli between the cerebellar hemispheres. So these are the three parts of that meningeal layer, okay? forms these special structures. So this is the meningeal layer of the dura mater, forms these three structures. Okay? And you can see them on this model. Now you guys do have a diagram that shows this, so when you are not here with our trusty models, you can use this diagram. It's basically the same thing as the model. Falx cerebri, this kind of cut edge here is the edge of the tentorium cerebelli. And then here, this little short piece is the falx cerebelli. Now notice there's one more thing you can see on this diagram and you can also see on this model. Notice all these weird blue stripes. In the diagrams and models, they always show deoxygenated blood in blue. Okay, it's not blue in the body, it's like a dark red, but they always show it in blue, so then you can tell the difference between the oxygenated and deoxygenated. Okay, so what these are is really kind of like large veins, and they're returning this deoxygenated blood that's coming from the brain that's going to go back to the heart. These spaces here where that blood is located are really between those two layers of the dura mater. Wherever those two layers, the periosteal layer and the meningeal layer, wherever they separate, we have these spaces that carry the deoxygenated blood from the brain, and it will eventually go to the heart. So this space right here, between the layers of the dura mater, is called the superior sagittal sinus. Sinus just means it's a space, okay? Superior sagittal sinus. I would like you to know that structure because we will see that this space is actually where cerebrospinal fluid will get reabsorbed back into the blood. Okay? So this space contains deoxygenated blood coming from the brain, but it also will be where CSF gets reabsorbed back into that blood. All of these spaces here are similar, and they all have their own names, but I would like you to know just this one for now, okay? Because that's where the CSF is reabsorbed. Okay, so let's talk about the other two layers of the meninges. Are you guys okay with the dura mater? We're just focusing on these layers around the brain for now. Around the spinal cord, they're actually easier, so I don't think you'll have any trouble with them. Okay, so this is what we just talked about. Okay, now the next layer of the meninges, going a little deeper, is called the arachnoid. You can say just arachnoid, or you can say arachnoid mater. Arachnoid means spider-like. This is a really thin, delicate, kind of wispy tissue. Pieces of it hang down, making it look kind of like a spider web. And it forms these little structures called arachnoid villi. These little structures stick up into the superior sagittal sinus. So eventually, cerebrospinal fluid will travel through these into the sinus. Okay, so let's just make a note of that. CSF will travel through them. Into the superior sagittal sinus. Okay?
So the direction is up and out rather than down and in?
Yes. This will eventually all make sense. We'll take a look at a diagram that kind of shows the whole pathway. Okay, and then the deepest layer of the meninges is called the pia mater. This is actually fused to the surface of the brain, so you can't remove it. It's on the surface of the brain. When you guys look at the sheep brain, it'll be on the surface. You won't be able to remove it. It won't look like anything special, but it's there. It's also fused to the surface of the spinal cord. Pia mater means tender mother. Tender mother. Okay, so those are the meninges. Let's take a look at the spaces between them. Okay. And then we will follow the pathway of cerebrospinal fluid, see how it goes from the ventricles into the subarachnoid space. So we're gonna talk about the spaces between the meninges. If you guys have heard of people who have maybe had some kind of head trauma and maybe have what's called like a subdural hematoma or a epidural hematoma, both of those can be fatal. So we'll see how these are actually around the meninges. And there are blood vessels associated with the meninges that if you hit your head just right, can cause a lot of bleeding. And it can actually happen very quickly so that the condition can progress so fast that, they really can't do anything for the patient once that bleeding, reaches a certain point. So it's very important. That's why it's important if you hit your head to go get checked to make sure you don't have bleeding into these spaces. Okay, so this is a frontal section. Just giving us a different look at these. This is the bone. Okay, so notice here we have two layers. These are the two layers of the dura mater. This outer one is the periosteal layer. It stays right up against the bone. The deeper one is the meningeal, and here it's coming down into the longitudinal fissure, forming the falx cerebri. Okay, the pink layer here is the arachnoid, and then this deepest layer, the one that follows the brain surface, is the pia mater. Okay, so you can also see them in this view. And notice here the arachnoid forming these little flower-like structures sticking up into the superior sagittal sinus here. These are the arachnoid villi, and that space, again shown in blue, is the superior sagittal sinus. Okay, so this diagram actually is, you can see all of that on this model. This model actually is for the layers of the meninges and the spaces between them. It shows you the flow of cerebrospinal fluid. Okay, so let's talk about these spaces. First of all, the epidural. Notice that the name actually tells you where it's at. You guys know epi means, yes, outside of. So the name tells you where the space is. It's outside of the dura mater. Now we said that the dura mater around the brain has that periosteal layer that's right up against the bone. But the periosteal layer is not actually fused to the bone. So there is a potential space there. Do you guys remember potential spaces from, I think it was like day one? We said that the pericardial cavity and the pleural cavity, all of that, those are potential spaces. Meaning that normally, the periosteal layer of the dura mater is normally right up against the bone, but it's not fused to it. So if something gets in there like blood, then the dura mater will separate and blood can collect in that space. So it is a potential space, okay, around the brain. Okay, meaning it has the potential to become an actual space. Around the spinal cord, because there's only one layer of dura mater around the spinal cord, this is an actual space. Okay, it's not a potential space, it's an actual space filled with fat, loose connective tissue, there's blood vessels here, there's nerves that travel through that space. This is where epidural injections are given, as you all know, very common for childbirth. And so the anesthesia is injected into the space. It diffuses below the level of the injection, so it will anesthetize or numb the lower body. Okay, so this is all about the epidural space. Okay, subdural. Again, notice that the name tells you where it's at. Sub means deep to. So this is deep to the dura mater. This is another potential space around the brain and the spinal cord. Okay, because normally the dura mater and the arachnoid are normally right up against each other. But again, if there's bleeding or some kind of fluid that collects there, it can become an actual space. And then finally, the subarachnoid space, okay, tells you it's deep to the arachnoid. This is an actual space, and this is the space that cerebrospinal fluid is located in. So when CSF leaves the fourth ventricle, it will enter this space. The fourth ventricle kind of dumps it off into the subarachnoid space, and it will circulate around the brain and around the spinal cord in that space. Okay, so let's take a look at what that would look like. You guys have any questions on any of these? And you can see these on this model, because again, if you look at this up close, you will see that it shows the meninges, okay? It shows both layers of the dura mater. It shows the superior sagittal sinus between them. It shows arachnoid villi sticking up into the sinus. It'll show the arachnoid and the pia layers. And it shows the subarachnoid space. Subarachnoid space is actually in purple here. So that's really what this model is for, all this stuff that we're talking about now. And notice that this looks just like this diagram, okay? Okay. So this is really for the flow of cerebrospinal fluid. And you can see here kind of, again, in one of those phantom views, like you're looking through the tissue, you can see the choroid plexus of the lateral ventricle kind of sticking up here. So we'll start with the lateral ventricles. We'll just trace the CSF. So it's going to go from the lateral ventricles through the interventricular foramina, which would be right about here, okay, down into the third ventricle, which is in this area. This is the diencephalon, and here's the choroid plexus of the third ventricle. Okay, and then it will go from there through that, oops, my arrow is off a little bit, through the skinny little cerebral aqueduct, which is this narrow little channel right here, into the fourth ventricle. And you can see how the fourth ventricle also has its own choroid plexus that it's showing you here, the red stuff. And then this is where CSF now leaves the ventricles and goes into the subarachnoid space. So everything shown here in purple is the subarachnoid space, and it will flow through that space, can go around the spinal cord and around the brain. Eventually, notice what happens. It's going to come around and go through the little arachnoid villi. And they will allow CSF to now enter the superior sagittal sinus, because the arachnoid villi stick up into the superior sagittal sinus. So once it enters the superior sagittal sinus, it just mixes with the blood again. It just becomes part of the blood plasma. And so it's made a complete circuit. It comes from the blood and it goes back to the blood. Okay, and all of that is written out for you here. For some reason, I don't know why, they started in the third ventricle. And really this should start in the lateral. So I would say you want to add something right in here. Okay, so you want to say, I would just put in lateral ventricles. It's going to go through those interventricular foramina. Okay. Now it goes into the third ventricle. So then you can just, again, follow it through here. You guys don't even really need this. If you just use your notes and the diagram, you can trace it yourself. Okay? But I would like you to know the flow of CSF. Any questions on any of that? Okay. So let's take a break. When we come back, we're going to go through some parts of the brain and talk in a little bit more detail about some nuclei and tracts that are located in different areas, okay? We won't go through the whole thing today, but we'll go through part of it, and we'll finish it on Thursday. Okay, let's come back at 8:45.